broken like that. But I like really need that time to get that immunology deck. It's about maybe an hour more work and then I'll get that posted. Okay, so I'll probably finish off this deck tonight and then start on immunology. So I apologize for that, it's just trust me when I say I needed it. Okay, so I think we left off on this slide on Thursday. We were talking about endospores, we were talking about the different components of bacteria. So I think it might be good to do a little bit of a review today since it's been a few days since we've had a lecture. Let's have a crack at some of these questions and just see how you do, okay? So, in terms of like the very basics, talking about prokaryotes versus eukaryotes, what are some of the differences that we talked about on Thursday? Yeah, the number one difference, right, it's in the name, prokaryotes do or do not have a nucleus. Do not, yeah, good. What are some of the other differences that we talked about? Is there that little table?
Reproduction, mitosis versus binary
Yeah.
fission.
Yeah, so he said mitosis versus binary fission. Which one is associated with prokaryotes?
Binary fission.
Binary fission. Right. Yep. What else? What else is different about them?
Organelles, the compartmentalization of the organelles is different. It's more like kind of like lazy in a prokaryote versus a eukaryote.
A little loaded, but yeah, sure. Yeah, so prokaryotes do not have as many organelles, and they probably are not as sophisticated as the organelles you would find in a eukaryotic cell. Absolutely. Do they have ribosomes?
One.
How- Yes, they do. Yes, they absolutely have ribosomes. What are ribosomes doing? Protein synthesis, yeah, they're translating mRNAs into proteins, so they've got to have those, but they are different. We'll talk about that more later, but that's one organelle that's very important that they both have. In terms of the genome, and when I say genome, what do I mean by that?
DNA cells.
Yeah, the DNA of the cell. So both prokaryotes and eukaryotes have it, right? It's how we encode our instructions for all those proteins. So when we're talking about the genome of a bacteria, of a prokaryote, what is different about that genome than a eukaryote? It's circular, yeah. Ours is linear, and we have, for us, we have chromosomes that are arranged in, with multiple chromosomes. We also have two copies, which makes us diploid. Bacteria are haploid, we only have one copy of those genes, so one circular chromosome. In terms of size, is there a size difference between prokaryotes and eukaryotes generally? The cell itself, which one's bigger? Eukaryotes are bigger than prokaryotes generally, yep. And with most of the clinically, medically relevant bacteria that we're going to be talking about, it's substantially, those are substantially smaller than eukaryotic cells. So I think those are some of the main things we talked about in terms of differences. For that second question, moving down this list, we talked about gram-positives, we talked about gram-negatives a little bit. So let's pick at one of these qualities about lipopolysaccharides, or LPS. You guys remember which of those two different types of bacteria we found LPS in? Gram-negatives, yes. And then we looked at the structure of that. So if you need to cheat and go back, by all means, look at that. But if you have it by memory, we talked about three different components of LPS, going from like outside on in. You guys remember, like, O antigen, yes, this antigenic O group. And then moving inwards from there, there was another component. Is that somatic O layer we kind of don't care about right now. And then that third component moving closer towards the interior of that bacterial cell was what? Lipid A, exactly. So that's the part we do care about. And why do we care about that? Endotoxin, yeah, but not so much release, not at least intentionally. It's integral, meaning it is built into that layer of the LPS. So the bacteria will keep it there structurally, but when bacteria die, they release endotoxin and that lipid A, and that causes problems for us. Okay, third question. We talked about capsule a little bit. what is it and what does it do for medically relevant bacteria?
It hides the antigens.
Yeah, antigen masking. Well, so what is it first? Let's review that. What is capsule?
I pictured like goo.
Yeah, it's goo. Somebody said the polysaccharides. Where is it on that cell?
Floating in the cells.
Yeah, it's on the outside. It's outside of the cell wall. So they have that big, thick, gelatinous layer of mucus that forms the capsule. And it masks epitopes. It also does what?
Phagocytosis.
It prevents phagocytosis. So those bacteria look like yummy little snacks for your immune system. They're covered in slime that makes it much harder for those phagocytes to engulf them and destroy them. Okay, and then fourth question, these are kind of random, but bacterial spores, why are they more resistant? What do they confer to those bacteria to make them resistant to the environment? What are the characteristics that enable them to survive? They become dormant, yeah. What does that mean to become dormant though?
So given the optimal environment, they can be alive.
They can, if conditions become favorable, they can germinate and become vegetative bacteria again. But when they are in that dormant state, what does that mean in terms of what's going on with their metabolically inactive? So it means that they're not replicating. They're not expending any energy copying their genomes and making daughter cells or reproducing at all. They also, in terms of the cell wall itself, is that the same or is that different than the vegetative cell? It's fundamentally different. They start expressing genes that change the character of the cell wall and ruggedize it in order to survive adverse environmental conditions. So then they can withstand higher temperatures, lower temperatures, drier conditions, desiccated conditions, higher pH, lower pH, different osmolarity, all sorts of like nutrient deprivation, those things. Those are the types of adverse environmental conditions that endospores will allow those bacteria to survive through. Do all bacteria have the capability of forming spores? No, definitely not. We only talked about, there really are only two medically relevant genera. You guys remember which ones?
Anthrax.
Bacillus and Clostridium.
CAs.
Yeah, okay. So you said anthrax. Anthrax is a disease caused by which bacterium? Bacillus anthracis. And the genus is what? Bacillus. Yeah, so Bacillus has members like Bacillus anthracis, it has members like Bacillus subtilis, Thuringiensis, a bunch of different members. And then that other genus we talked about was Clostridium, which has members like difficile that we've talked about, or we will continue to talk about, Perfringens, tetani, which causes tetanus. Okay, so those are two very important genera that we'll talk about later. Structure, you guys want to play a little matching game? Okay, all right, we'll start with one. So let's look at this long whip-like thin appendage that's coming out of the terminal end. What is that? A flagella. Rudabaugh, yeah, okay, good. What about number two? It looks like we're looking at a shorter appendage. Pili, yeah, exactly. So that is a pili, singular pilis. We talked about the pili being responsible for a type of activity bacteria can do.
Is it like wrapping cells?
It's interacting with them.
Conjugation.
Conjugation, bacterial conjugation. So cell A, donor cell, reaches out to cell B, recipient cell, forms a pilus, oftentimes called a sex pilus. And that is it's like a tube that allows DNA basically to travel through that tube from donor to recipient. And that is done by these pili. Let's look at number three. It looks like we have some shorter, very numerous hair-like projections all around the cell. What is that? Fimbriae. Yes, very good. Number four, this is a little tricky to see where that arrow is going, but looks like that yellow layer outside the cell. What do you think that would be? Capsule. Yeah, capsule. Is that associated with gram-negative or gram-positive? Trick question is both, right? Both. And not all bacteria produce it, some do, some don't, but it's agnostic of whether it's gram-positive or gram-negative. So 4 is capsule, very good. Let's move a little bit further inward. What is this orange layer? What would that be?
Cell wall.
The cell wall, exactly, that's the cell wall. Do we as eukaryotes have a cell wall? We don't, we have a cell membrane, but this would be unique to bacteria. But this is what makes gram-negatives gram-negatives and gram-positives gram-positives. They have different cell wall characteristics. So we'll go over that again too, but that's the cell wall. And then interior to the cell wall, this is number 6, this is getting really hard to see, but on the inside of the cell wall, then you have the cell membrane. Cytoplasmic membrane, the plasma membrane, same. Plasma membrane. Alright, what else do we have left? Number 7, we're looking at that thick sap on the interior of the cell. Cytoplasm, yeah, exactly, cytoplasm. That's where you have a lot of this enzymatic activity. ATP is at least the synthetic pathways that produce ATP that can then go to the plasma membrane and generate respiration activities. All that is occurring, all that metabolic activity is happening in the cytoplasm. And those dots are just indicating that it has ribosomes so they can do protein synthesis. And then last but not least, it looks like we have some binary fission going on. There's a septum that's forming between these cells. So number 8, what are these white patches that are forming? That's the DNA, exactly. So there's no nucleus, but it is organized a little bit. It's called a nucleoid, but it's not the same as a nucleus. So, but that is where the DNA is, that's where the genome is, in that kind of semi-organized configuration in that cell. Okay, and then last but not least, let's hit bacterial shapes real quick. There were some names and some vocabulary that we went over to talk about these different shapes we use in the lab a lot. So when you see a bacterial cell on a gram stain, looking, through my microscope, and you're seeing it, if you see them and they look like spheres, what do we call that shape? Cocci. Is that singular or plural? Plural. Yeah. What's the singular? Cox. Or coccus. Yeah. Staphylococcus. Yep. Let's say you see something that's more cylindrical or rod-shaped. What would that be called? Bacillus. Yeah. And then the plural of bacillus is bacilli. Good. What about if it's almost spherical, but it's a little bit shortened? Coccobacillus, yes, very good. This is, I don't have an example in this cartoon, but we talked about a club-shaped morphology that's kind of like swollen on one end. Cariniform, dipteroid. And then I missed one, I didn't indent that, but there's another shape associated with a curved, comma-shaped morphology. Vibrio, yeah, that's a Vibrio shape. This would be like a Vibrio example. And then 2 more very similar but different spiral-shaped types of bacteria. One was flexible, one was rigid. The flexible one was the one that had the flagella built into it, endoflagella. Spirochete and so it's hard to tell this cartoon, but yeah, spirochete and spirillum. Spirillum was the tight corkscrew one. And this last one is not even on this slide also, but if, just as a term, if we're talking about multiple different shapes of the same species bacteria, it's pleomorphic, meaning it can have different appearances depending on environmental conditions. Bacterial arrangements, this isn't in the form of a question, but just to kind of review. We have singles, pairs, and clusters, chains, tetrads, palisades. With palisades, do you guys remember what that looked like? Kind of stacked next to each other. Yeah, rods stacked next to each other. Okay, so that's just a quick review from Thursday. Any questions about that stuff? Alrighty. So we are going to talk a little bit about some lab processes. The stuff we're going over today is much more general than what we'll cover in block 2, so we'll hit it much harder. This is still pretty superficial, just to kind of orient you, especially if you've never worked in a microbiology lab or seen how bacteria are cultured or other pathogens. So, just as an intro slide there, you can see somebody working with a pure culture with an agar plate. Oftentimes if you're working in a clinical lab, you have a nice big stack of plates waiting for you in the morning when you come in to work up and work through. But what is the purpose of doing this? Like, why would we bother to go through all the energy and effort to culture and bacteria?
You can do culture sensitivity, so you identify what's growing, and then you can do a sensitivity to see what bacteria, antibiotics are sensitive to it.
Yeah, exactly. So it'd be all academic if it was just identifying the bacteria. Who cares, right? Except for the fact that that has an effect on what treatment you use for the patient, right? So in order to get that information, you first have to know what bacteria it is. Then you can do antibiotic susceptibility testing and figure out exactly what antibiotics that particular bacteria is sensitive to. So that's kind of the name of the game. It's why we go through all this hassle of cultivating these in the lab. In terms of a few terms to be familiar with, these are things that kind of help you get an understanding of different types of bacteria and what we can expect for their metabolic needs. Not all bacteria that are clinically relevant are specific pathogens for humans. Bacillus subtilis, for example, it's a soil bacterium. It does fine without a human host. And so its energy requirements are going to be pretty general. It does well on a lot of different types of media. It's easy to grow up. A lot of bacillus bacteria are pretty easy to grow. And those will consume, they live out in the environment because they're able to break down and metabolize non-living organic matter, soil bacteria. So most of these are, a lot of bacteria are like that. Those are called saprophytes, eaters of basically dead material. Another term to be familiar with is fastidious. If bacteria are fastidious, what do you think that means for our ability to grow them in the lab? They're very picky. Yes, it means they have very specific requirements for growth. They might need extra sources of iron. They might need iron presented in an easier way, different vitamins that other bacteria wouldn't need, different atmospheric conditions. So picky or high maintenance bacteria, they're difficult to grow. Parasitic bacteria, in this context, by parasitic bacteria, I mean bacteria that absolutely need a host in order to survive. Those can be pretty difficult to grow too, especially if they are intercellular bacteria. Intercellular bacteria don't do well outside of cells. So when you want to just grow them on some media in the lab, it can be challenging. Sometimes you actually need to have living cell cultures in order to cultivate some of these bacteria. So things like chlamydia, some of the rickettsias that we'll talk about later. Those are obligate intercellular bacteria, and we oftentimes don't even bother cultivating them in the lab. We'll do a PCR or something else, or an immunoassay, because they're so challenging to culture. Okay, so in terms of like the reason why we do this, again, we already talked about that. We need to understand their characteristics, which might differ from strain to strain, and we need to get a definitive idea on what these bacteria are before we can do antibiotic susceptibility testing, figure out how to treat them appropriately. Most of the time, when we receive specimens in the clinical lab, we're going to plate them on solid agar and grow them up there. You can also grow them in liquid broth as well, but usually you're only doing that for specific reasons. So with these agar plates, if you've never seen one before, it has like an alginate base, and there is a little bit of moisture in there, so it's not a dry substrate. There's some water in there to keep them alive, but then they also have a ton of nutrients. They have carbon sources and nitrogen sources. We'll usually put in like yeast extracts and peptone extracts, so that allows bacteria something to survive off of. In terms of the specimens that we're actually plating on these agar media plates, what are some specimen types that you think of that would be coming from SIP patients?
Urine,
Like a specific specimen type, like what part of the body, for example?
Throwing a bugle swab maybe?
Yeah, it could be potentially a buccal swab, if somebody said blood. Anything else?
urine,
Urine, like, we could go on and on, stool, CSF, spinal fluid, synovial fluid, wound swabs, wound specimens. So in the micro lab, we usually have our cells arranged in what we call benches, and benches are specialized parts of the lab. There's a urine bench, those guys deal with like a tremendous high volume number of urine specimens for working up UTIs, pathogens that cause urinary tract infections. The blood bench is a busy bench, the wound bench is busy, the stool bench, et cetera. So all these different types of benches will have different types of media that are selected, that are designed to capture the bacteria that we would most likely expect from those specimen types. Once we plate those specimen types, whatever they are, CSF, wounds, whatever, you gotta leave them alone for a while. This is like a traumatic thing for these bacteria. They're used to happily dividing and doing their thing in the human body. You take them out, you collect them on a swab, or they're already in liquid like CSF or whatever. You gotta transport them down to lab, and then the lab needs time to receive them and plate them and then put them on media. That's traumatizing for the bacteria. So there's a time sensitivity there. You need to be pretty quick about getting these specimens plated on these different media types. And then you stick them in an incubator that has the right atmospheric conditions for the right amount of time. You give them time to recover and start replicating on that media again. It has the right pH, it has those right nutrients, right salt levels, all of that. And then depending on the type of bacteria, those bacteria will reproduce enough where you actually get visible colonies growing up, like you can see down here. So this top plate, we talked about different morphotypes of colonies a little bit. You can see this is a mixed culture. There are many, many different types of bacteria on there. You don't work up a plate like this. You have to isolate one of these colonies that you think is medically relevant, and you subculture it. So like down here, this to me looks like Serratia marcescens. It's that orange colony. It's a nice pure culture that you can work up for identification and antibiotic sensitivity tests. In terms of how long it takes for these colonies to form, again, it depends on the bacteria, but we talked a little bit about how long a generation time is for bacteria on average. You guys remember how long it is on average? 20 to 30 minutes, yeah. So some of the faster ones might be as little as 10 minutes before they start dividing Clostridium perfringens, 10-minute generation time. Some of them take 12 to 16 hours, now we're talking about 20, 40, 36 times longer to culture those and grow those. So the implications there are that microbiology lab has to have their SOPs written to hold these plates long enough where you might be able to cover some of those slow-growing bacteria, like Mycobacterium tuberculosis, very slow-growing bacteria. But most of these are going to grow fast enough where you plate them in the afternoon, you come back the next morning, overnight growth, and you might actually start seeing some colonies. Those colonies, theoretically, those came from a single bacterial cell, and they replicated exponentially until you have hundreds of thousands, maybe a million different clones, basically, of the same bacteria. Those are colony-forming units. So that's what we're looking at there. Like hundreds of thousands of millions of bacteria that are so densely grown up that you can actually see them very easily, visibly with the naked eye. And from there, we'll take one of those colonies, put it on a glass cover slip, look at it under the scope after we've stained it, and get some preliminary information about what type of bacteria is in that colony. So that's the basic gist of it. I don't think this slide adds a whole lot to that, except for we see another example, nice pure colony. Same, these are pretty much all the same bacteria, nice pure colony. Same species on a different type of plate. And this allows us to see a different feature of this bacteria. In this case, what kind of energy, what kind of metabolism it has, what kind of sugars it's fermenting, and this acid byproduct is changing the color of the media. So that's all useful to the people in the lab. And then, as I mentioned, you do have some liquid broth options, and that can give you information about, like, is it microaerophilic? You can look at the turbidity and see how densely these things are going, but again, oftentimes we're more reliant on that solid media type. And then going back into the incubator, when we have our specimen plated out and ready to put it in the incubator, these are some of the conditions that we're careful of. Not all species of bacteria do well under normal atmospheric oxygen conditions. In the human body, we have pathogens that do well, like deep in the intestines, which is basically an anaerobic environment. Those bacteria, they might be able to tolerate oxygen or they might not. Some bacteria are what we would call microaerophilic, like Campylobacter and H. pylori. So we have to grow them up under reduced oxygen conditions in order for them to survive. Other bacteria are capnophiles. H. pylori is also a capnophile. That means that it likes to have a relatively high percentage of carbon dioxide. That's just what it's used to, it's what it grows best under. The bacteria that cannot tolerate oxygen at all are anaerobes. We have strict anaerobes that basically will die upon exposure to oxygen. They don't have the enzymes that they need in order to detoxify oxygen, which can be a pretty toxic molecule, draft an oxygen species and a bunch of other, and free radicals, for example. So those bacteria, they do not do well, and you need to grow them under anaerobic conditions in order to culture them. The vast majority will be aerobes, though, so they'll be fine. Like it's a good beneficial advantage for bacteria to be able to tolerate oxygen. So most of them are going to fall into that category of being aerobic. They like their set humidity levels. They don't like things to get too dry. So if you get under 60% humidity, those bacteria start suffering a little bit. They don't do as well. And finally, for temperature, most incubators are set around 35, 36 degrees, some are 37 degrees. What temperature is a human body? 37 degrees, exactly. Some of them might like a little higher, like Campylobacter. We incubate that at 42 degrees. But most bacteria will do fine at around body temperature in the incubator. In terms of growth, so we have, again, we've verbally talked through this, but when you start plating these bacteria out, you can have very few viable bacteria in that specimen. But under the right growth conditions and given enough time, they will have this exponential amplification of cells until you get these visible colonies form on the media plates. And let's say you've let them go for 24 different cycles of generations, 24 generation times, about 12 hours, let's say. Theoretically, you could have 16 million bacterial cells from one precursor progenitor cell, right? Can that trend continue indefinitely? Definitely not, right? We'd be living in a world of bacteria. So there's something has to give. So we can only grow them up to a certain level. They start running out of nutrients and they start dying. So those will die off even under the best growth conditions. So they start dying and we'll have a reduction in the number of colonies. We've talked a little bit about intracellular pathogens before, but this is sort of underscoring what we were talking about the difficulties in culturing extracellular and intracellular pathogens. Most of the bacteria that we're talking about in this course are extracellular bacteria, things like Staph aureus, a lot of E. coli are extracellular, fairly easy to grow for the most part. But when we're talking about intracellular pathogens like Rickettsia or Mycobacterium, Chlamydia, Coxiella, those guys, again, like because they need all these extra cellular factors, they tend to be more difficult. So nothing new, we haven't really said anything that we haven't said before, but just know that intracellular bacteria tend to be more difficult to cultivate in lab. And because of that reason, again, if we can help it, we'll generally use other techniques for identifying bacteria like chlamydia and rickettsias and those guys. On that note, some of those weird ones fall into this category that kind of lazily gets referred to as atypical bacteria. With atypical bacteria, we're talking about things like Rickettsia, Anaplasma, Ehrlichia, Legionella, These have generally a very small size, considerably smaller than regular bacteria. They don't stain well with a gram stain for various reasons, but usually has something to do with their cell wall, characteristics of their cell wall that we'll also talk about more in block three. And some of them, like mycoplasma, even though we defined prokaryotes as having a cell wall, mycoplasma do not have a cell wall at all. They're still bacteria genetically, but they have more like a cell membrane with sterols in it. So needless to say, those are weird, they're atypical, they don't stain well with gram stain, they don't stain at all with gram stain, they're clear. And in terms of treatment implications, we can't use antibiotics that we would use to target the cell wall. So things that would be inhibiting peptidoglycan synthesis, for example, would not work on these atypical bacteria. We also have a whole category of bacteria called acid-fast bacteria. They definitely have a cell wall. In fact, they're super rugged cell walls. But because of how waxy they are, these cell walls, a lot of very lipid-rich content, lots of mycolic acids, they don't stain at all. With normal gram stains, you have to first acid treat them or heat treat them in order to get them to take up a stain. And that also makes it very difficult to treat patients with these as well because of that cell wall. Okay, let me see. Let's knock out fungi and then we'll take a 10-minute break. You guys doing okay? Okay. All right, so fungi. These are eukaryotes. We talked about on Thursday a couple different types of pathogens that fall into the category of eukaryotes. So this is one of them. This is in kingdom fungi. You guys remember two other kingdoms that we talked about that are eukaryotic kingdoms? Animalia and Protista. Yeah, exactly. So fungi are in kingdom fungi. Eukaryotic cells, they have a cell wall, they have a nucleus. Their cell wall is very different than ours though. They have lots of glucans, they have other carbohydrates that we don't have. They have weird sterols and this type of like ergosterols, type of cholesterol that we don't have in our cell membranes. They have chitin, which is another type of carbohydrate, one of the most abundant biopolymers on the planet. So different cell wall, but they have a cell wall and they do have a nucleus. In terms of forms that we see, both of these can be forms that we will find in pathogenic fungi. But yeasts are unicellular, so you can see on this micrograph, the skin electron micrographs and yeast cells, basically pretty spherical. They can gram stain, so they'll take the gram stain, so they look like bacteria, only bigger, a little bit bigger. And these guys, when they are dividing, they will divide by budding, so or binary fission, much in the way that bacteria do. But they do, again, they do have a nucleus, so this is mitosis that we're talking about. In terms of size, again, like bigger than bacteria, 4 to 15 micrometers. Most bacteria we said were somewhere in that ballpark of like 1 to 2, 3 micrometers, so noticeably larger. With molds, these are fungi that are multicellular. They have filamentous hyphae that we'll talk about in the next slide a little bit more, but those hyphae are pretty long, I think up to 30 micrometers long. So that is helpful in distinguishing them. And then on the ends of these hyphae, so this stalk is a hyphae, a reproductive hyphae, they will have these unicellular spores. So those are the circles that are on the top of the stalks right there, those are the spores. And those are what those molds will use to reproduce. The spores will aerosolize and germinate into new molds. In terms of the yeast, this is what they would look like if you looked at them microscopically. We have some budding yeast there. When they bud, they have a very broad bud base, called broad-based budding, hard to say. And then you can also culture them on agar. So they look a lot like bacteria, but these are yeast cells that are growing up as colonies on agar. Usually you have a pretty shiny appearance. And in terms of the length of time it takes for them to grow, a little bit longer than bacteria, generally speaking, 2 to 5 days to grow up those yeasts. You guys ever heard of Candida or Candida albicans? That's an example of a yeast-only fungal pathogen we'll talk about a lot more. With the molds, we have, we're probably most familiar seeing these kind of benign innocuous molds that will grow on food, right? But we do have some serious fungal pathogens that have mold forms as well. When you're looking at these mold structures, you're probably all familiar with, ever notice that kind of fuzzy, hairy appearance of those? That is because of their structure on a microscopic level. If you were to zoom in on that fuzziness, what you see are hyphae, which the hyphae is called the mycelium. That's like a visible mass of hyphae that you see. And they have that kind of feathery appearance because of the way those reproductive hyphae are extending out and they are growing all those spores on their termini. So that's what gives them that appearance. They have a very different morphology than yeasts. When you grow them up and you culture them, they take longer to grow, 5 to 21 days. And if we look at the structure of those hyphae, multicellular organisms, right? So these are specialized cells. The hyphae are either vegetative hyphae, which means that those are the hyphae that are extending out into the agar or to whatever substrate and extracting nutrients from them. It's what's keeping those organisms alive. And then we have reproductive hyphae, which are again another specialized form of hyphae that are responsible for growing upwards and supporting spore formation. Those spores themselves have a lot of different appearances. You can see here some like spherical spores that are forming on the tips of these reproductive hyphae. Those are called microconidia. We'll talk more about that later. And then look how different these are. These are still conidia that we're looking on the right, but those are macroconidia from a different species of fungi. So that's kind of like its own little specialty, mycology. People are very good at reading those and identifying what they look like based on morphological structures, but that's basically like mycology 101. Okay, any questions on bacteriology or mycology? All right, I've got 10:40, let's take a 10-minute break and we'll pick up on the slide.
From what it is, it's like it's already mostly very sophisticated with that.
Yes. I got the Institute coin, but I didn't get the company one because she had
What do you think
it.
about the wireless bus?
I get one from Capital.
But
But I know she did say she's gonna be here in. She said she's gonna be in December. She's gonna bring us. Yeah, right.
Really?
Yeah. She's told me something that.
There's that video.
She was just like hit or miss.
Yeah, I think she really
Like,
because
Way too.
he studied
I know she's a good
history of
person.
the 5 or 6 math distinctions we've had. One of them is kind of associated with transition from dominance from cold-blooded animals to warm-blooded animals. Part of the theory there is that cold-blooded animals aren't first set.
So you're in those markets, that's my volunteer service.
The rise and success of what would otherwise be relatively weak warm-blooded animals after that mass extinction wiped out a lot of the reptiles on the planet. Yeah, it's fun job, just not adapt. They do have countries that can grow at 27 degrees, but they tend to do better. That's separates off the environment, break it down.
Do I have a really good one?
That's 25 degrees, that's a pretty healthy place. So when you put them in a warm-blooded animal, allow them to go
Like that break. In the sense of like I feel like go back into study versus like.
yeah, that's the fear. That's the fear that you have with adaptation occurs, that the whole pathogen or potential pathogen suddenly able to thrive.
I thought that was
And you also kind of say
same thing like really well done. Don't be mistakes.
don't give people shoot to see actually do retaining about studying.
But pretty accurate from the standpoint of microbiology.
In terms of that concept, yeah, it's like, it's very, you know that scene, so whatever it was, there's
You know, yeah, normally like I want like it's all like
like
what's happening now.
the index of things that happen in Malaysia,
Is there anything I'll study
and they're
so
doing
far?
like an autopsy.
Oh, yeah.
And they call in this expert, she's somebody big, she has this epiphany of how bad it is, and she's like,
Side day for about like whatever. Yeah, so
or it's too late, or whatever she said.
whatever time I
But,
have that day I'll study a little bit.
That's your open issue.
And then Sunday is more like
Yeah,
study.
the whole point is that she can have realization
Next Sunday I'm gonna do grass here.
adaptation to this.
What are the possibilities of what happens?
Well, like in that show, probably not good. So cordyceps is actually a genus of fungus that it affects ants, right? Yeah, exactly, insects. And all those scared things that you see in the show, like behavioral modulation, post causes them to basically lose their essences as human beings. That happens sort of to ants that are infected, right? They become, like zombified. Or the gene just and then they die slowly because the fungus goes in the brain. But like in terms of how realistic that would be for that, I am more skeptical about. But it's kind of like a hodgepodge of different theories from the mass extinction theory and what we actually see with cordyceps and prior ants.
Crazy.
Yeah, it is interesting.
Want to get your opinions on it?
Love that show. Haven't seen season 2.
Is that how?
You have to pay for it.
To the micro concentra which builds their powder.
One thing we could fluctuate just to like.
Just using the remember like
But I remember it just. We didn't get it. But
always a
a single hit out in the blood.
Yes, well, in the sense that
Okay, let's get going then. Before we get back into the deck, just for funsies, take a second to go through that really cool graphic. The link should be on your slide deck if you wanna play with this, but you know, we talk about size a lot, size of these different organisms. So this is a cool little tool from University of Utah, I guess, that puts into relative perspective the size of some of these microorganisms relative to normal objects that we are used to seeing with the visible eye. So kind of orienting you to this graphic, in the upper left they have this grid, and what we're looking at first is within a 1 millimeter size range. So obviously like super easy, we can see coffee beans, grains of rice, 12-point times regular font, like a little bit harder to see are sesame seeds, but still easy to see. And then down here, grain of salt. So zooming in a little bit, still easy to see grain of salt. That's 0.5 millimeters. What's kind of considered the limits of the visible range would be when we get down to, I don't know, 0.5 millimeters, 0.2, somewhere in there. So this amoeba proteus, for example, which is 0.5 millimeters or 500 micrometers. Yeah, I don't know if you'd be able to see that. It's right on that limit. Below that, we've got, cursor, yeah, okay, cool. Paramecium, that's a type of protozoan or protista, slipper cell, little aquatic protist in that 200 micrometer range. And then we have our first human cell, which is the human egg, ginormous cell, but not visible to the human eye probably. So that's on the range of about 130 micrometers. We zoom in a little bit more, we can see a skin cell, a fibroblast, that's about 30 micrometers. And that's a pretty good sized cell in terms of like what we see in the human body, so that's not a small cell by any means. In the eye, we have those photoreceptors, rods. Those are pretty long, up to 100 micrometers. Looking a little bit closer, there's sperm, tiny little sperm. By width, that's only 5 micrometers wide, so that's about the width of that bacterial cell, but obviously pretty long. And then a tiny little red blood cell, look how much smaller a red blood cell is than a fibroblast. It's 8 micrometers. The size of an X chromosome, 7 micrometers. Baker's yeast, this is a fungal cell, a fungal yeast cell that I mentioned, Saccharomyces cerevisiae, Baker's yeast. Remember that. Zooming in more, finally we get to see some bacteria. There's E. coli with this long flagella, maybe some fimbriae around it, and that size range of around 3 micrometers by width of that cylinder. Mitochondria, a eukaryotic organelle, a little bit bigger than the size of E. coli. Another organelle of eukaryotes called a lysosome, which is a compartment that's responsible for degrading cellular garbage, debris. And we zoom in a little bit more, and we start getting into the range finally where we'd be looking at viruses. So we're gonna, first couple slides of the virology portion, we're gonna look at size ranges as well. But measles, this is a paramyxovirus in the news a lot these days. It's a kind of a big virus, 220 nanometers now, so 0.2 micrometers. So that's on the bigger end of the scale for viruses. Smaller than that, we're getting into influenza, 130, HIV, also kind of a moderate sized virus. And then these teeny tiny viruses, these are on the lower end size rise. Hepatitis viruses, doesn't say which one, there are multiple different hepatitis viruses, but 45 nanometers is pretty small. And then rhinovirus, which causes, you guys know what rhinoviruses cause?
Like cold.
Yeah, it's one of the many common cold viruses. So that's really tiny, 30 nanometers. And then we have some more organelles of eukaryotic cells. We've seen ribosomes before. You can see them on electron micrographs easily. They look like grit in a cell in cytoplasm. So ribosomes are on the size range of 30 nanometers. And then smaller than that, they start getting on a really tiny scale where you can only see structures like antibodies, these globular proteins with advanced techniques and advanced microscopy, electron microscopy, X-ray crystallography, things like that. Hemoglobin, oxygen carrying protein. And down to the level of methionine, which is, what's methionine? It's an amino acid, yep. Adenine, that's a type of nucleic acid, a nucleotide. All the way down to a single carbon atom, which is 340 picometers, 0.34 nanometers. Alright, so it's kind of a cool little graphic, and I think, if you wanted to reference that, I think it's helpful. Okay, so a good segue to start talking about viruses very superficially. What are they? We mentioned this when we talked about taxonomy. They're sometimes defined by what they're not more than what they are. They are not living. They do not metabolize anything. And when they are inside of a cell, they are entirely reliant on that host cell machinery in order to replicate. But that's not metabolically active. That is able to reproduce viral proteins in the viral genome. So with bacteria will grow and divide. A parent will divide into two daughter cells, but when a virus infects a cell, it produces many, many copies. So the exponential growth is a different growth curve. In terms of what it can infect, this is not one size fits all. Different viruses have very specific host ranges. Some can infect animals and people. Those are called zoonotic viruses. Some of them can only infect humans, and some of them can only infect certain tissues in humans. So that's called tropism. And we'll cover that in more detail later too. In terms of their genome, which we defined as the nucleic acids in an entity, to use really broad terminology, viruses can have DNA genomes. It's still a genome. It's still the nucleic acids of that entity. But they can also have an RNA genome. And that is funky and different, and no cellular life can exist like that. Only viruses can have that RNA genome. So they can have one or the other, but they can't have both. You're either a DNA virus or you're an RNA virus. Not surprisingly, if you were to treat a viral infection with some antibiotic, you would probably not have a lot of success, because based on what we've talked about so far, you know that a lot of the targets in bacteria would not be what you would find in viruses, right? You're not going to be able to hit them with their cell wall, something that's targeting their cell wall, or cell membrane, or ribosomes, because they use the host cell for all that. So it makes developing antiviral medications much more challenging. We do have them, and we'll talk about them in block 4. We have antiviral medications, but it is a different ballgame when we're talking about treating viral infections versus treating bacterial infections. And that's part of the reason why it's so important to actually identify what the etiologic agent of a disease is. If somebody comes in with respiratory symptoms, is it a viral respiratory infection or is it a bacterial respiratory infection? It would have a lot of implications for treatment. This is what I was talking about with the size, so probably not impressed by that, but we saw the red blood cell all the way down to teeny tiny SARS-CoV-2 mid-range. Too small to see with microscopy, right? The limits of microscopy, you wouldn't be able to see viruses, so there's no point in taking a sample and putting it on a slide and looking at it under a standard light microscope like you would do for bacteria. You can see them with electron microscopes. This is a transmission electron micrograph. You can see see like HIV particles budding off of an infected host cell. But that's very expensive, it's very time consuming, and we have better, faster diagnostics for confirming viral infection. So in this course, I'll show you tons and tons of electron micrographs of viruses, but that's not useful for diagnostic purposes. In terms of the morphologies, most of these viruses are going to be spherical. Talk about how much they vary in size range before. A couple notable exceptions, like rabies virus, for example, Rhabdoviridae. Those are defined or described as having like a bullet-shaped morphology, kind of distinctive. The filoviruses like Ebola and Marburg virus, also extremely distinctive. They can have this appearance which is described as filamentous, meaning wire-like, or they can be practically spherical. It just depends on the conditions in which they're budding in the host cell, but pretty distinctive. Variola virus, anybody know what that causes or what that's associated with? Smallpox. That is, which is no longer circulating today, fortunately, but we do have other pox viruses. That has a little bit of a distinctive shape, and it is a massive virus, 360 nanometers, so that's above the normal range for viruses. Really what you need to know on this slide is like the typical size range for these viruses. 20 to 300 nanometers is important. And that's not something you see with regular light microscopy. That's the main point there. In terms of structure, when we're talking about the genome, as I said, it's only RNA or DNA. And these funky little things, they can be single-stranded or double-stranded. We're talking about DNA in eukaryotes and prokaryotes. Is it single or double-stranded? Always double-stranded DNA. That's our genome for cellular life. Viruses, some of them are double-stranded DNA, but they have this weird ability to have single-stranded DNA genomes as well. And for the RNA viruses, you can have double-stranded RNA, which if you're covering that, are you guys covering that in biochemistry yet, or are you talking about nucleic acids? Today? Cool, okay. So when you talk about nucleic acids today, we'll talk about it in more detail in block 4, but think about how weird it is to have double-stranded RNA. They can do that. And they can also have single-stranded RNA, which is more common. So they have this delicate little teeny tiny genome, which doesn't usually encode for that many genes. They're not that elaborate. Anywhere between 7 on the lower end up to a couple hundred for the bigger viruses. But compared to us as humans, we have 30,000 different genes, give or take, and genomes that are billions of base pairs versus maybe 20,000 nucleotides. So again, like the tiny little genome, they need to be able to protect that genome. So they usually will have something like a capsid covering that genome. The cartoon on the top there is showing a protein capsid that is covering that viral genome. And those proteins are encoded by the virus itself. So the host cell isn't encoding that particular protein that's coming from the viral genome. It has to be expressed and translated and then assembled together. But that's a viral origin. And its purpose is to protect that genome so it can infect new cells. Other viruses, they will still have some sort of protein covering to protect their genome. Like you can see here, kind of like a helical genome, but those large spheres are nucleocapsid proteins that are protecting the genome. And down here you have another capsid. But then this outer covering there, that's an envelope. It's a lipid bilayer envelope. Where do you think that virus gets that? From you. Yeah, from the host. Because what do you have? Your cells are covered by a plasma membrane, a phospholipid bilayer. So that's exactly what the virus takes from you when it buds from one of those infected cells. So 2 different flavors, either non-envelope naked capsid virus or envelope viruses. Another little game for us to play, a little matching game. So let's look at 2 different structures. We're looking at, as a spoiler, one's capsid and the other is enveloped. which one is the envelope virus? One on the left or the one on the right? Good, yes, envelope virus. Which one is the capsid virus? And we'll call that naked virus, same. Hang on, let me see what I put next to this. Okay, on the envelope virus, this arrow up here that's pointing to that yellow, which one of these terms would that be? Don't you? This yellow thing on the right, on the envelope virus, what structure is that? It's the envelope, exactly, that is the envelope. I should have planned this out a little bit better. Capsid, would that be pointing to this, the inside, or that structure in the bottom there? I'm sorry, this is terrible magic. The bottom one, yes. So capsid is the entire exterior shell of this capsid virus, but it's also present in this envelope virus protecting the genome. So on both of those. And then we only have two left. We've got either viral receptors or nucleic acid. Of those two options, this top one pointing to the exterior of this the virus particle, which I think it is. Viral receptors, yes. So the virus needs to have those receptors in order to enter some host cell. And then lastly, our nucleic acid is like a cartoon in the middle there. Sir.
Just to make sure I was right, so all viruses in the genome have a protein shell called a nucleocapsid, right?
Yes.
And then in classifying the exterior viruses, one is classified by capsid shell, capsid viruses, and then the enveloped virus, two types of classification.
That is correct. So he said, in terms of classifying viruses, you have a protein nucleocapsid covering the DNA or RNA genome of all viruses, and that is true. You do have a nucleocapsid to protect that. And then in terms of the ultimate exterior, you have one of two different types. It's either enveloped or it's capsid. But yeah, exactly right. And those capsid viruses, unfortunately, they do have a couple other names, like you'll hear non-enveloped or you'll hear naked. It's all the same. Okay, I apologize for this figure. It is one that I've carried over from other instructors. I like it because it shows both DNA and RNA viruses, and it contrasts the differences between them, but it tends to confuse people a lot. So I do apologize for that. This is an artificial thing. You're not ever going to have likely the same four target cell get infected simultaneously by a DNA virus and an RNA virus. But we'll walk through it because we need to talk, at least in an overview, about the steps in viral replication. So let's have track of that. So you are a virus particle. You're looking for a host cell to infect. You're on the outside of that host cell. In order to infect, you have to have that specific binding interaction that lets you know you're targeting the right host cell. You need your specific virus, you need these specific things. So viruses like SARS-CoV-2, that's what we're looking at in the center here, that blue virus particle. Those viruses will have glycoproteins or receptors on their surfaces. Those are exactly what they sound like. They are proteins that have sugars on the outside of them, glycoproteins. With SARS-CoV-2, I'm glad that we use that as an example in this slide because everyone knows this virus so well. We call that glycoprotein spike protein. It's what goes into all your vaccines that you guys have been getting for years now. So that spike protein will recognize a specific cellular receptor. You've probably also heard of that before too. It's ACE2. And it has like a bit of a handshake when it encounters cells that express ACE2, which is a very widely expressed receptor for angiotensin II. What that looks like is you have a SARS-CoV-2 spike protein, and this is the protein globular form. You can see in purple here, that's the spike protein, corresponds right there. And the part that actually touches and has that specific protein-protein interaction with ACE2 is called the receptor binding domain, the RBD. And then in green, that's the host ACE2 receptor. So that binding interaction is very specific. SARS-CoV-2, it can use other receptors like neuropilin-1, for example, but this is a specific receptor that it looks for in order to infect those cells. Once it has or makes that specific contact, like on the right here, it's making that specific receptor contact, it enters the cell, in this case by receptor-mediated endocytosis. So this receptor just thinks it's handling angiotensin II, and it internalizes that entire virus particle into the cell. It doesn't know any better, it just recognizes protein motif. So it takes this virus into the cell, and then you basically have the ability for that virus to hijack the rest of the cell and make copies of itself. So we've already attached, we've penetrated into the cell. With the case of SARS-CoV-2, that's receptor-mediated endocytosis. Other viruses will enter by membrane fusion, viruses like HIV, for example. And then once you're inside the cytoplasm of the cell, you have that covering over your genome, right? What's that covering called again? The nucleocapsid. That's got to come off in order for you to start transcribing viral genes and reproducing or replicating that viral genome. So that's called uncoating. The nucleocapsid comes off, and then you have the genome laid bare. That genome has to be, the genes in that genome have to be expressed into mRNA transcripts. Those have to be translated into proteins, viral proteins. And then the genome itself has to be replicated. So all those synthetic events have to happen, and all of those synthetic events are mostly relying on host cell machinery in order for that to happen. When you're done making copies of that genome and viral proteins, it's time to put it all together again. The host cell will do a lot for the virus, but it won't do that. The virus is responsible for self-assembly. So it has proteins called matrix proteins, will self-assemble all these components together, and it will package them into nearly complete virus particles that are ready to be released from that host cell. When those are ready to be released, if it's a capsid virus, if it's a naked virus, whatever, usually those will accumulate in huge numbers in that target host cell until that cell basically bursts or lyses, and then it releases all those virus particles en masse all at once. When you have an envelope virus, the method of egress is much gentler. It's basically budding. You have that, it's going to come out with an envelope, right? So we have a diagram that will show this in a second, but it basically will just gently bleb out of that plasma membrane and absciss or completely bud off from that target, and it'll go float off. With those viruses, that can happen with virus particles leaving individually. So over time, you have virus particles blending off gently. So it's like death by 1,000 paper cuts. The cell won't even notice one virus particle leaving with a little bit of its plasma membrane. But when you have hundreds leaving or thousands, that's when the cell starts to feel the burden of what's happening to it, and you get cytopathology. All right, so fairly briefly, let's start with what's happening with the envelope DNA virus on the right-hand side here. Mostly focus on the fact that this is a DNA virus. Okay, it's coming in because it has an envelope. In this example, it is fusing with the plasma membrane, and it has that nucleocapsid entering the cytoplasm. It will uncoat, and then its genome has to go into the nucleus. If it's a DNA virus, that means it's going to use the host cell DNA replication machinery, which is in the nucleus, right? So it will go into the nucleus, it will make, it will get transcribed, those genes. Those mRNA transcripts that are transcribed have to be exported out into the cytoplasm so they can be translated by what? Ribosomes. Yes, ribosomes are out in the cytoplasm, so they have to go out into the cytoplasm, get synthesized into viral protein. And meanwhile, the genome itself is being replicated in the nucleus by those host cell nucleic like DNA polymerases. Once those viral proteins are ready, like I said, they come back into the nucleus, and you have assembly of those little virus particles, like the core virus particles. Those will get assembled and then moved to the plasma membrane, trapped up to the plasma membrane, and then, like I said, they kind of just gently get released from the cell at that point. With these RNA viruses, you do not typically have any involvement of the nucleus because it's an RNA genome. All of this can happen out in the cytoplasm. So when they come into the cell and they uncode that little piece of RNA, same, it will get transcribed if it needs to be, but viral proteins will be made ultimately by those ribosomes that are out in the cytoplasm. And the genome will also get copied. Once you have the genome and those viral proteins, same thing. You start assembling together into viral particles, and then you leave the cell. If it's a capsid virus, like shown in this example, that's a much more violent kind of bursting of the cell when it leaves. Okay, and here we see a little bit more detail. We're talking about an envelope virus, how they enter the cell. You have an envelope that is docking. Viral receptors on the envelope are docking with specific receptors on the host cell. Those membranes are fusing together, and then the genome is getting dumped into the cytoplasm. When they're leaving, when envelope viruses are leaving or budding from the cell, a similar process happens, but kind of in reverse. You have that mostly assembled progeny virus, it's coming into contact with the plasma membrane there, it's causing the plasma membrane to deform, and basically, once it gets to this point, it reseals the cell, and that virus particle is released. Okay, so all of those things, from the time that the virus first enters the cell to the time you have all those progeny virions released from the host cell, that takes time. Different amounts of time for different viruses. Some of them can accomplish all of this in as little as an hour. Some of them will take 20 hours to do this. But during that phase, when you have your initial exposure to a virus, and those viruses are trying to find appropriate target cells to infect, that takes time where you have pretty much no symptoms at that time because the virus is busy doing all these biosynthetic events. Once they get released though, like I said, it's not just 2 viruses for every 1 that enters. That's not the right output ratio. It's like hundreds of viruses for every infected cell. So you get super exponential release of virus particles for every round of replication. Just depends on the virus. Some of them produce a lot of progeny, some of them very little. But at some point, just like when we talked about with bacteria, you don't continue to have an infinite number of replication events and cells that are infected, right? Your immune system steps in for one thing, and it will start just kicking the shit out of those viruses and stopping this exponential growth at some point. So this will stop, and you will have some decay of viral progeny. Those levels will start dipping down at some point. But very important point here, it is variable. This is why we study viruses separately, independently, because they have different kinetics of producing progeny variants. Okay, with most viral infections, and I think this is the last slide for, and second to last slide for viruses, but with most viral infections, these are acute infections. The viruses go into the cell, they do their business, and they, as quickly as possible, make progeny virions. Some viruses, though, have the ability to establish latent infections in cells, which means it's good and bad for the virus. Good in that they don't attract a lot of attention from the immune system. They're not exposing themselves much. They get into the cell, they transcribe very little, if anything, from their own genome. They're not replicating a lot, and they can kind of hide out that way for weeks or months or years. Viruses like herpes viruses, for example, those are latent viruses. HIV can establish latency. So yeah, they can hang out there and avoid detection from the immune system for a very, very long period of time, potentially forever. However, at some point, there could be some sort of environmental stress or cue, something like you become immunodeficient, you lose your ability to fight off infection, you become sick, you get antibiotic treatment, something like that, some trigger, and it leaves you vulnerable to what we call reactivation for those latent viruses, which means they start becoming biosynthetically active again, transcribing and translating, expressing genes, building new virus particles. So we'll talk about that, some examples of those later. And then lastly, in terms of how we can identify these in the lab, we can culture viruses, but we don't very often. We do it for research purposes. Clinical labs usually don't do this anymore though because it takes such a long time. Anybody know what we rely on mostly for identifying viral infections in the lab? PCR. Yeah, PCR is kind of, or immunoassays, which we'll talk about in block 2 as well. For research purposes, if you want to grow up virus, or even in some clinical labs, it works basically by using cell lines as an indication of specific cytopathological effects. Different viruses cause different cytopathology. So if you take a cell culture, this looks like a cell line of fibroblasts, it's the monolayer of those fibroblasts, and you take a specimen from, let's say, like a respiratory swab, somebody who has a respiratory virus, inoculate that within days, 2 to 28 days, whatever. Eventually those cells will start having some pathology, and they usually end up dead. In this case, you can see the cells have detached from the monolayer, they're all warped, beaten up, dead, apoptosis, just completely destroyed. So that used to be the way that we would identify viruses in lab, but these days we seldom do it. Okay, let's see, 120. Are you guys doing okay or dying? Doing okay? Do we need like another short break? No, okay. Okay, we'll drive on then. There's not that much left in this deck, but looks like you guys are kind of suffering a little bit. So I apologize for that. But we'll drive on and talk about prions.
Question back here.
Yes, sir.
There's this slide before this one. Did you take it out?
What's
Bacteria.
Oh, yeah. Yeah, I think I'm just gonna table that until next block. Yeah. Yeah, good question. Okay, so prions. How many of you guys are familiar with prion diseases? A couple of you? Like 2 out of 65? Okay, so what prion diseases have you heard of before? Mad cow disease, chronic wasting disease, yeah. Anyone else think of any other prion diseases? Well, it causes bovine spongiform encephalopathy in people, yeah. Yeah, so those are some well-known prion diseases. Regardless of which ones we're talking about, they are exceedingly rare in humans. The reason why I sent out that article about chronic wasting disease is, I think I mentioned on Thursday, is because that is becoming a concern increasingly, particularly with hunters, because of what happened with mad cow disease, which was a disease that affected cows in the UK in the 90s, and it caused a lot of deaths because people were eating meat from cows that were infected with mad cow disease, and it wasn't cooked to the point where it was destroying these infective misfolded proteins. So that got a lot of attention, and it caused, as a consequence, a lot of additional regulations about how we handle meat and export criteria and a lot of things. But we worry about that with chronic wasting disease here in North America because an increasing number of deer and elk populations are testing positive for chronic wasting disease, exhibiting the same symptoms that cows did in the UK, where they start stumbling around, they lose their motor control, and that was transmissible to humans. With chronic wasting disease, so far it isn't, but it's one of those kind of like nightmarish scenarios that we worry about, that someday it will adapt and be able to become infectious in humans. And in terms of the nature of these particles, what they are, they're a little mysterious, probably still understudied a little bit, but they are unlike any other thing that we're just talking about in microbiology, topic-wise, and this is the last time we'll talk about them, so you won't see them again in any of the other blocks. Because they kind of defy categorization. They're not organisms, they're just proteins. They are hard to detect, they are hard to diagnose, they are pretty much impossible to treat. It's tricky to understand how they're transmitted. So a lot of big questions out there, which is why they are so scary. But What they are basically is this prion protein, which is a normal protein in your brain that becomes misfolded. The nomenclature for that is PRPC, C means cellular prion protein. That's the normal one. When it becomes misfolded, it gets this little SC designation on the end, which stands for Scrapie, which is another type of prion disease that affects sheep. So for the transmissibility, it's kind of a mystery how you can have a misfolded protein, which sounds like it would be an isolated problem. But what we think happens is this misfolded protein comes into contact with other prion proteins, and it causes them to misfold also. So you get an amplification effect, and that's called conversion. You have misfolded protein contacting other prion protein and inducing that same misfolding. The diseases that they cause are generally called prionopathies. We have a lot of prionopathies in animals, and we have some in animals as well. But the common feature is once you have a kind of transmissible, propagating prion issue in your brain, that is ultimately fatal. You can't do much to reverse that. And once you start exhibiting symptoms, it's usually pretty rapidly fatal. So it can take years, a very slow build when you exhibit symptoms, but once you become symptomatic, you usually die fairly quickly. Those symptoms, they're kind of what you would expect with something that's destroying brain tissue. You would have things like loss of coordination, psychiatric issues, sleep disturbances, things like that, dementia. And again, since this is your own protein, you're not going to be triggering the immune response, and you can't clear these misfolded proteins. With acquisition, we have this very mysterious category, sporadic, where we don't have any idea what's causing it. We have genetic or familial acquisition of prions with like Creutzfeldt-Jakob disease and some other prion diseases, where this gene is acquired and inherited from one generation to another. This gene that will cause a misfolded prion protein. And then most famously, we have the acquired form, where you come into contact usually with brain tissue and acquire the disease that way. We are increasingly recognizing that this could be a problem in hospital environments, especially when you have surgical equipment that's been used on come into contact with brain matter. If it's not sterilized completely in an autoclave, that could carry over infectious material. So a lot of like lab and hospital SOPs are written with concern for prion transfer. It's super scary. So laboratory and clinical, we talked about eating meat, right? Mad cow disease that was caused by just eating meat. Kuru is another one that's pretty famous, if anyone's ever heard of Kuru before. That was kind of a crazy one, associated with cannibalistic practices in Papua New Guinea, a specific tribe that had a cultural practice of, like when their loved ones died and passed away, they would eat a large part of the body, particularly the brain. And because of that, you had like this weird high prevalence of Kuru disease in that population until the 1980s or so, where I think they finally were like, yo, stop eating brains. It's causing problems. Satisfied folks postulates, and they're like, don't do that anymore. But it was like much higher prevalence in women, because women were the ones that were processing the brains and tending to like digest it more, I think. So yeah, Kuru is pretty much no longer a thing. You don't see that anymore. But it's definitely an acquired form of prionopathy. Going back to the pathology, this is sort of what happens is a normal brain tissue. But over time, when you start getting an accumulation of that misfolded protein, you get damage to the brain, neuronal damage and degeneration. So these different forms of prionopathies like Kuru and Scrapie, which is again only in sheep, not a human disease, and classic Creutzfeldt-Jakob disease. What you're looking at is spongiform encephalopathies, these weird pockets that are forming in brain tissue that destroys the brain's ability to function properly. And then lastly, the last thing we'll say on this is just going back to this point about zoonotic risk. Scrapie is one of the best characterized in animals, Scrapian sheep. It gets that name because it causes sheep to behave erratically, so they start scraping against, like they start scraping their heads against whatever, and they'll do that until they get significant damage. Anyway, they get the name scrapey from that, from scraping their heads. Aberrant behavior, stuff like that. The bovine spongiform encephalopathy is that one really well-characterized example of an animal phenopathy that is transmissible to humans. So that's the one that we have worried about and responded to significantly. And then again, chronic wasting disease is one that, as far as we know, is still restricted to those ruminant populations, but we have a lot of that in North America. Okay, if there are no questions on prenopathies, I'll just move on to parasites, and that's the last piece of this deck. Okay, you guys okay? Okay. Perfect. All right, so protozoans. Moving back to eukaryotes, we talked about these being in Kingdom Protista, so kind of jumping all over the place, but nucleated pathogens and parasites, they get classified according to how they move around. Amoeba we're probably familiar with. It's amoeboid movement is simply like that, like wild flinging out of cytoplasm basically. These extensions called pseudopods that allow amoeba to move around by just like throwing out projectiles and crawling around that way. We have flagellated parasites as well, things like Giardia. If you've ever heard of Giardia before, but it has like 8, maybe 9 different flagella, 4 on each side, and a central flagella that it uses to just kind of float around. It's an enteric pathogen, so it stays in your gut most of the time. And then you have ciliates. What are cilia? Yeah, little extensions. So ciliated protozoans, they'll just sit there and kind of beat their cilia and move around that way. It'd be like the euglena that we saw. Yeah.
What's the difference between cilia and fimbriae? Is there a big difference?
Well, fimbriae, that's like a bacterial specific feature, and that's used for attachment specifically. So bacteria would use fimbriae, fimbriated bacteria would use those fimbriae to facilitate attachment. These guys, yeah, these protozoans that are ciliated, they use that for motility, just to get around and beat those cilia. Like in your respiratory tract, you know how you have cilia lining your respiratory epithelial cells. The purpose of those mucociliary cells is to beat up pathogens up and out of your respiratory tract. So same deal here. They're just actually that's a ciliated parasite right there. You can see a very fine layer of cilia on the outside. They're just beating those in unison. And then lastly, we have the P. complexa parasites. Be things like malaria here. These are gametocytes from Plasmodium species. They don't have a lot of motility at all. If they have any, it's sort of like a little bit of a body flexion, but they're unicellular, so it's just like one cell that they'll like maybe wiggle around a little bit, but there's very little motility with the P. complexa. Things like, again, Plasmodium for malaria, Babesia, everything like that. In terms of size range, 2 to 50 micrometers. This is well below the visible range, so definitely need the aid of a microscope to see most of these. But they're big enough where you can see them with irregular light microscope, most of them. Some of them get quite large, but not the clinically relevant ones. For the morphology, especially when we're talking about amoeba, we usually are dealing with 2 different stages. There's a cyst stage and a trophozoite stage. Both stages are pretty much necessary for most of these protozoans because they encounter different environmental conditions. When things get rough, like let's say you're an intestinal protozoan and your host, like, I don't know, is sick or has a course of antibiotics and conditions become rough. So you form a cyst, you get defecated out into the environment as a cyst, and you need to stay there until somebody, you enter a water source and somebody else drinks you up and you can go back into a nice friendly environment of someone else's gut at that point. When conditions become favorable like that and you're back in your optimal niche, then that trophozoite form is this more amoeboid form right there. And that is when you can do things like eat can replicate. That's what the name trophozoite means. It means an eating animal. So conditions are favorable, you're a trophozoite, you're usually a little bit bigger. These are easier to identify most of the time. And then the cyst stage is when things are usually unfavorable. You need to survive. So metabolically not very active, not feeding, not replicating. In terms of reproduction, most protozoans can replicate asexually pretty well, which has the advantage of being something you do very quickly, very rapidly. Through binary fission, much in the way that bacteria will do binary fission. Some of them have more complicated asexual reproduction called schizogony, like malaria, Plasmodium parasites do that. And that will, I think we'll wait until we get to block 5 to discuss its schizogony form. Some parasites are able to sexually reproduce as well. Malaria is another example where it can do asexual and sexual reproduction. So some of them can undergo conjugation, and then this other form called syngony, which is when the gametocytes will fuse together. The Zookeeper complexans, again, that's like Plasmodium and Babesia species. Very, very common. This guy at the bottom right here is, I should ask, anybody have a guess what parasite that is? No. Okay. Has anybody ever heard of Giardia before? Yeah, old man Giardia, it's one of those intestinal protozoans we're gonna be talking about. So this is a trophozoite of Giardia, and when it's ready to reproduce, it basically undergoes binary fission, which just splits right down the middle. And look at that, it looks like a heart, doesn't it? Isn't that sweet? That is sweet. Alright, so those are unicellular parasites, our protozoans. With our worms, another word for worm is helminth. These are multicellular. Huge, tremendous diversity in them, but most of these are going to be, they're parasitic, they're going to be adapted to acquiring nutrients at the host expense, which means a lot of them are going to be in the gut. Some of them are tissue parasites, but yeah, most of them are going to be intestinal. These 3 main groups are what we'll be concentrating on in block 5 for parasitology. Tapeworms, which are cestodes, flukes, trematodes, the common name in the more technical term, and then roundworms, which are nematodes. All of these helminths will have complex life cycles and transmission cycles. It takes a long time to study them. They're very detailed. With these cestodes or tapeworms, these are flat worms where their entire body basically is segmented into these different portions called proglottids, which are basically reproduction machines. Each one of these proglottids is nothing but like hermaphroditic ovary and testes together. So in that same compartment, they just sit there and self-fertilize themselves and produce eggs, this entire body length. In terms of nutrient acquisition, they kind of absorb nutrients. They don't really have a digestive system per se, so they absorb nutrients through their skin, which is more accurately called a tegument. But that's basically cestode anatomy. They do have a tiny little head up there. That head doesn't have any kind of nervous system, but it is used for attachment. So it's going to attach to the intestinal epithelial cells, but the rest of the body is just like flapping in the middle of the intestinal tract, acquiring nutrients and reproducing, generating eggs. In terms of size, these adult worms, if they're in your body and they've been there for a while, like they set up camp for a while, years and years, the longer they're in your intestinal tract, just doing their thing, living their best cestode life, the longer they grow. Some of them, like the fish tapeworm, can get to be up to 10 meters long, so it's like, this is from me back in the classroom, just incredibly long tapeworms, but it does take time. So 25-year infection, 30-year infection with tapeworms, the host is minimally impacted, hardly even knows it's there, but they're quite lumpy. And others are much smaller, so. Trematodes, these flukes, they are non-segmented, still multicellular, but non-segmented. They have a little bit more of a sophisticated digestive tract than the cestas that we talked about, the tapeworms. They do have, like for example, it's called a blind gut, where the mouth is basically, like the entry for nutrients is also the exit for nutrients. So I guess if the timing of eating has to be carefully thought of if you're a trematode. One way in and out. Exactly. They're definitely macroscopic. I have some great images of flukes in the intestinal tract for block 5, so we'll see. But some of them, like the giant intestinal fluke, can be quite large. And you can have them, like imagine what leeches look like on the outside of the body when you've gone into leech-infested waters and you come out covered with leeches. They get that way. They get heavy infestations in the gut as well, where if you do like a colonoscopy of a patient with a heavy fluke burden, you can see flukes all over the intestinal tract. And they are attaching kind of similar in the manner that tapeworms do. They have an attachment point which injures the epithelial intestines, it's like epithelial cells as well. So you can have a little bit of anemia from that injury, but mostly they're just sucking up nutrients over time. So that's the majority of the pathology with trematodes. These guys eat monoecious or dioecious, which in this slide I didn't put a name on that, but monoecious just means that you have both male and female. Most of the time they are monoecious, with the exception of schistosomes, which are dioecious, means they have both males and females, separate genders. Lastly, I think, yes, last slide is dead. Nematodes. They're round words. They are, I guess, highest on the evolutionary ladder. They have the most complete digestive tract. They have a beginning and an end, yay. And they take in nutrients only through the mouth. So they're not messing around with their tegument anymore. They're just taking in nutrients through that oral cavity. These guys can get to be fairly large. Ascaris lumbricoides, for example, is the one that's probably most famous for intestinal nematodes. That can be about the width of a pencil, and you can get heavy infestations of those guys. They always are gendered, so dioecious, there's always males and females that are reproducing sexually in your digestive tract or wherever they are, and producing generally like huge quantities of eggs. So as you are nurturing these adults and they're generating a lot of eggs, those eggs are getting released into your stool, and then that's the specimen you send off to the lab. They do an OMP, an ovarian parasite exam with a microscope, and very easy to see these parasitic eggs with a microscope. Easy to identify as well. So that is it, unsegmented next to that. Any questions about that quick primer for parasitology or prion diseases? Okay, I will go and finish off that immunology deck, and I'll get it posted by this afternoon, and then that'll be the rest of this block. So I'll sit down and go from here. Okay, thank you guys. Let me know if you have any questions.